Good afternoon, everyone. Welcome you all to our webinar this afternoon. We have two dynamic speakers with us today, Dr. Michael Toews and Dr. Tracy Leskey. Dr. Toews is a primary research entomologist with the University of Georgia, and his responsibilities in applied insect ecology and pest management in row crop system. He has a BS degree in biology from Fort Hays State University, a master's degree and a PhD in entomology from Oklahoma State University. Ecosystem and co-teaches two graduate level classes. He's a native of Wilson College and a master's degree in ecology from Pennsylvania State University and a PhD in entomology from the University of Massachusetts. So you can see from today we have two dynamic speakers. And at this time, we're going to turn it over to Dr. Toews for the first presentation.
Okay, good afternoon, friends. As Willie mentioned, I'm with the University of Georgia. I do want to point out that many of the pictures that are in my slide presentation this afternoon are taken from our bugwood.org website, and so that's something that you all could use as a resource anytime you're putting something together. There's several thousand freely available pictures there. The other thing I'll point out is that my slides are available as a narrated slide set on the protectingus.now.org website, and that's under the tab called Educational Materials, and so all the slides that I'm going to show today you can grab from there. Today we're going to be talking about a relatively new invasive insect pest started here in the southeastern U.S. Kudzu bug is a rather malodorous little critter, as we'll soon find out, that it occurs in extremely large numbers, and its range is rapidly expanding across the southeastern U.S. and marching into other parts of the country. This particular insect is originally native to southeastern Asia. We see it in China, India, Indonesia, Japan, Korea, Malaysia, Pakistan, Sri Lanka, Taiwan. It's also been described from a few specimens in Europe and parts of Africa. This insect started in 2009 in metropolitan Atlanta, where we saw very large aggregations that were occurring on light-colored houses. And it's interesting that that was the first observation, was not a agricultural citation, but rather an urban nuisance, and we'll see that how that's kind of played out. The taxonomy of this thing has been kind of difficult. Obviously, when you have a completely new family, it takes a little bit of time to get the taxonomy straightened out. And then we went back and looked at the literature from other parts of the world. There were a lot of pseudonyms that were being used, even down to the common name, bean plataspid, the lablab bug, the globular stink bug. But the scientific taxonomy wasn't all that clear either. Megacopta punctatissima was commonly used, as well as what we're terming our bug, which is Megacopta cribraria. There are a number of different scientific names that have been used in other countries, and we really had to coalesce in terms of, is this the same bug or not? I'm going to use the term kudzu bug throughout the presentation today, but I do want to point out that kudzu bug actually has not been accepted as of today as the officially recognized common name with the Entomological Society of America, but a petition for that has been submitted, and we expect that that's going to happen in the near future. So the current range expansion of this insect is really one of the things that's gaining it a great deal of notoriety. So you'll see that in 2009, we have confirmation right up there in the metro Atlanta counties. And from there, then within each year, we have very rapid range expansion. You see that we're covering most of North Georgia within 2 years. And interestingly, those specimens in 2009 were just in the fall, so that was just in a few months there. So 2010, we've got rapid expansion that we actually have counties in Alabama as well as South Carolina. And you see in 2011, we made large increases up towards the mid-Atlantic part of the country. And each year, we've been able to add to that as this thing marches forward. So as of today, I believe that we've got observations in 13 different states, 582 counties, as well as the District of Columbia. In terms of the known reproductive hosts of this particular insect, there's really two that are of importance to us here in the Western Hemisphere. The primary reproductive hosts are kudzu and soybean, and that's where we see very large populations that build up. And when I say reproductive hosts, those are plants that support populations all the way from egg to adult, so they can develop on those plants without any other sources of nutrients. I would also put in there in our tests that edamame and pigeon pea also support, but those are very closely related species to soybean. Now, in terms of things that we've looked at, kudzu bug spends time on a lot of other different hosts, and we put that in parentheses. We looked at things like pinto beans and winter beans, chickpeas, fava bean, snap bean, lima bean, black-eyed peas, the list goes on. And in all of those, the adults will hang out and do okay, but those really don't support development from egg to adult in the same way that kudzu and soybean do. So in terms of what I'll term non-reproductive hosts, or places where we find large aggregations of these bugs just hanging out, man, that can be just about anywhere. And so I've got some pictures here from my plots, and I'm down in the southern part of Georgia, but there's no doubt that if I have corn or wheat or cotton, peanuts that are adjacent to my soybean plots, I will always find kudzu bugs in those other crops, but none of those support development. In fact, we think they're just hanging out there, especially in the spring when the populations are moving around. We see kudzu bugs in those crops. And then probably the most obvious answer or case there would be on the telephone pole. I assure you that the bugs are not feeding on a creosote-soaked telephone pole. Nonetheless, you see large aggregations there, and that just sort of proves our point that these things are hanging out at different places, especially in the spring while they're waiting for kudzu to green up. The bugs are also commonly observed in different parts of the urban landscape, and that's been problematic for us at times. Okay, this bug was actually first observed as an urban nuisance as opposed to an agricultural pest, and these populations can be particularly difficult to manage. So in the different pictures here, you see groups, for example, there on the right on a white porch. It looks like light colors are really attractive to this particular pest. And from a pest management perspective, this is challenging for us because any place you have upright white surfaces, we tend to aggregate bugs at certain times of the year. And what our pest management professionals and exterminators have found is that, yeah, they're easy to kill with an insecticide, but that does not provide lasting suppression because you're really just killing a very minuscule number of bugs relative to the overall population, and those areas tend to get reinfested pretty quickly. So what we're recommending now is if you give them a few days, they're going to move on in search of a feeding site. The exception to that rule is if you have known reproductive hosts that are close to a house, for example. So if you've got kudzu growing in the backyard, you're going to have a constant source of insects, and really the alleviation for that condition is to remove the kudzu as opposed to spraying the bugs that are actually lighting up on the house. In terms of the human health concerns, we need to establish right from the get-go here that kudzu bugs cannot bite humans or any other animal. Therefore, we're not concerned whatsoever with any type of disease transmission. So there's no biting that goes on there. Kudzu bug mouth parts, are really only good for piercing green plant tissues. But the images that I'm showing you here come from a couple of years ago. One of the first years we had, in fact, it was the first year I had kudzu bugs in my soybean plots. I decided I'd go ahead and scout in shorts one day, and the bugs were falling down between my boot collars and my socks, and with every step I was literally squishing kudzu bugs and kudzu bug immatures. You can see from the pictures there, I was squishing the bugs and the hemolymph came out, and that caused some chemical burns on the skin that I would characterize as extremely itchy. It was kind of like having poison ivy on you. That lasted for about a week. You can put some anti-inflammatory topicals and things like that, but that's not something I would recommend. That was actually a pretty miserable week. And since then, we've noticed that if you have a kudzu bug that lands on your arm when you're out in the field or in town, if you smash them, you get the same thing. So there's a very definite sort of a chemical burn almost that takes place anytime that you smash those actually on your arm or on your neck or something like that. This insect has proved to be really challenging for us, not just from urban and agricultural standpoints, but it has some international trade implications as well. In the spring of 2012, Honduran officials discovered dead kudzu bugs in a shipping container from Georgia, and they ended up stopping all inspections, or to step up inspections of cargo containers from the Southeast: Georgia, South Carolina, North Carolina, Tennessee, and Alabama. At one point, we had nearly 4,000 containers being held up in Honduran ports, and so you can imagine the problems that causes when we're shipping perishable commodities. In addition to that, we've established that kudzu bugs are attracted to tall, upright surfaces, and we also discovered, or Honduran and Guatemalan officials intercepted live kudzu bugs in the passenger areas of commercial airplanes coming from Atlanta, as well as the baggage compartment. And so in response, our USDA APHIS folks have had to work with the airport authorities to develop new protocols to mitigate those problems. Now we've talked a little bit about how serious a pest this is and some of the places where it's had economic impacts. Let's talk a little bit more about the bug's biology, ID, and pest management. So kudzu bugs have piercing, sucking mouthparts, and they work only on plants. They feed on the stems and the petioles of the kudzu or the soybean plant. And those infested plants are characterized by dark-colored purplish lesions on the stems and petioles, and that's what's going on in the bottom picture there with the yellow arrow. So this is similar to aphid honeydew. That excess liquid that's being shunted through the insect eventually is excreted out, and it lands on the plant exterior where it creates a favorable habitat for secondary pathogens like a sooty mold or something like that. Unlike our native stink bugs and some of our other imports like the southern green stink bug, this particular pest does not feed on developing pods. In terms of the types of damage that kudzu bug causes, specifically looking at soybean in this case, they do really affect our yield. And so if you were to, let's start by looking up at the picture in the upper right. In the right, I've got some example soybeans from plots that were protected using insecticides, and you see that those pods are great big and full size. In the unprotected tray there to the left, you see a lot of much smaller beans, and the problem there is as the nutrients are being shunted out of the plant and through the bug, it doesn't allow for the nutrients to get to the pods to fill the beans. And so we see not only smaller beans, but really unfilled pods, fewer beans per pod, and of course much smaller beans, and all of those contribute to yield loss. So we feel at this point that kudzu bugs are really just a stress-inducing pest on soybean growth. On average, we're looking at losses around 20% at this point, but in various trials that's been higher. We've had several that have been up in the neighborhood of 50%, and in the most extreme case, we had a 60% difference between the treated and completely untreated plots. One other thing that we see is greening around the edges of the field. That's notable down there in the bottom right. In that case, the soybeans that are really heavily infested don't senesce down to become easily harvestable by machine. In terms of the life cycle on this critter, we're looking at 2 generations per year, and the developmental time varies a little bit based on the season. The spring generation, that's going to be in an April to May time frame, generally takes about 8 weeks. We've got some cooler nights and daytime lows in there, so it slows down. Then the summer generation, which is a June through August developmental time, that's generally going to be about 6 weeks, and that's just due to warmer conditions. All true bugs undergo incomplete metamorphosis, so the immature stages are termed nymphs. We've got 5 different instars here, and the immatures have a similar diet to that of the adults. They do look similar, although they've got some long white hairs. I'm showing you with the arrows on the right. And the 5th instars do have some obvious wing pads. In terms of the eggs themselves, they tend to be about 1 millimeter long. The most obvious thing there is that they're laid in rows of 2. So they're about 1 millimeter long, about 0.5 millimeter wide. They're generally very white immediately after oviposition, but then they change to a pale salmon color with some dark bands going the length of the egg. And those are laid in 2 parallel rows, and they have that obvious ring of spines around the operculum open up. Adult females deposit a small brownish particle on the underside of the egg mass. That's what we're showing there with the blue arrows. These unique structures are called symbiont capsules, and they contain a specific symbiont bacteria. Newly emerged nymphs will then actually ingest those symbiont capsules, and that facilitates transfer of the gut symbiont from the female to the new progeny, and that enables them to feed on a much larger assortment of diet. The first instars remain very, very close to the egg cases. So if you're dealing with first instars, they're going to be right there. Then as they get a little bit older, then they start wandering about the plant. The external wing buds will develop. You can see there on the far right in the fifth instar. The later instars kind of range from a green to a brown as they move about the plant. The nymphs are also gregarious, just like the adults. And they typically do develop on the same plant where they hatched. Identification of this insect is unique. They are the only member of the Plataspides, so that's a new family for the Western Hemisphere. And they're really distinctive because they have this large shield that we call the scutellum that covers the entire body. That's unusual in stink bugs. So that scutellum actually covers everything. There's a kind of a suture there that we're showing with the red arrow. And then in terms of separating the sexes, if you look down on the truncated posterior, females tend to be fairly rounded. So you get kind of a rounded posterior end of the abdomen. Males are very truncated. It's almost a linear cutoff, and that's really obvious when you've got a specimen in hand when you're separating males from females. These are very different morphologically from the other stink bugs, be that southern green, or brown stink bugs, or even brown marmorated stink bug. We don't have that triangular appearance that we do with most of the stink bugs that occur in North America. Hibernation and dispersal is an area that we've been doing a lot of work, trying to understand exactly where these things are hanging out. Kudzu bugs hibernate as adults, so we have fall dispersal of the second generation adult, and that really correlates with the most urban complaints. Bug populations from senescing plants in the fall are really looking for areas to get away from the cold weather, and the most common area that we find here in the southeast to be underneath pine bark, and that's what I've got up there is some pine trees that I was pulling apart in January and February. In late February, on a warm sunny day, you might actually see them out, and they look like they're just sunning themselves, and then they go hide back underneath the bark of that tree. Then they begin to leave that overwintering area in March, and they're dispersing in our part of the country anyway to developing kudzu. The first generation occurs on kudzu, and then that generation, once they've developed, then they start looking for vegetative stage soybean. In terms of monitoring this insect, they're attracted to just about any kind of a light-colored upright surface. So while we don't have a pheromone at this point, it's really not necessary because they're just attracted to light colors. And if it's tall and upright, that's going to work just fine. So here's two examples of traps that we've developed. The first is a cardboard vein trap that's just over a black bucket filled with soapy water. The bugs hit the vein and then they'll fall into the soapy water where you can count them. The trap on the bottom is inexpensive 3-inch PVC, and I've just wrapped a piece of fly or stable fly paper around there, and they just get stuck on the trap, and that allows us to sample populations over time. Okay, in terms of monitoring in soybean, traps haven't proved particularly good in soybean, so what we're using there is a sweep net. Preferred method right now is 10 sweeps in a single row. This is some images out of a video that I put together earlier. These populations can be very, very large. We've seen as much as 100 bugs per sweep. So at really high populations, and that's typically in areas where the bug is invading for the first time, the numbers can get extremely large, and that's the kind of situation I went into when I had them falling into my boots. And they will fly into your hat, into your ears, just about anywhere. This is kind of a not so pleasant insect to deal with under really, really large populations. The sweep net does a very good job sampling for adults. It does bias you more towards adults than immatures, and we feel like one of the triggering pieces for determining a threshold needs to be the presence of immatures. So we're recommending that we actually do visual inspection of soybeans as well. You can sweep for the presence of the bug, but once it's been determined that the bug is there, then we need to be looking for the presence of immatures, and that's really best done with a visual inspection. So on the right side there, you can see those bugs, and the immatures really blend in very well with the vegetation. So you need to look closely to see those green, brown, and olive-colored insects. In terms of a treatment threshold right now, we've got a lot of work that's underway looking at different types of thresholds and whether they're appropriate growth stages. What I would say right now is that we are very sure of is that if the immatures are not present, spraying tends to not be economically wise. What happens if you spray vegetative stage soybeans is that the plants get reinfested very, very quickly. Now, they're easy to kill with an insecticide, but during the vegetative stages of soybean growth, we don't seem to be suffering any kind of a yield penalty. So we need to wait until the immatures are present, and that tends to be a little bit later in the year. I'll show you a little bit of data on that as we move on. Okay, in terms of insecticides that we're using, they are pretty easy to kill with most broad spectrum type materials. Any kind of pyrethroids, say a bifenthrin is very efficacious. Any kind of organophosphate like acephate or any of the carbamates like carbaryl, those all are very effective. Insect growth regulators like diflubenzuron, as well as any of the lepidopteran specific materials have not proved to work well. So things like spinosad, flubendiamide, and doxycarb, as well as any of the newer diamides, those really do not provide us with adequate control. Interestingly, we've got some exciting developments on the biological control front. This insect is actually not a serious pest in Asia where it's native to, and the reason we think that that's going on is it's under good control of egg parasitoids. So there's been a lot of work in this part of the country looking at different native egg parasitoids, and we looked at many different species, and none of them attack this particular species. There have been some isolated flies, a couple of tachinids that have come out of some parasitized stink bug adults, as well as some general predators, say the spine soldier bug or the Florida stink bug that will feed on kudzu bugs, but none of those appear to be exerting any kind of control. And so we continue to look for other things, and just last year, an egg specialist, Paratilenomus saccharalis, showed up here in Georgia, ironically in the same area where the bug was first discovered. And as it turns out, Paratilenomus is indeed one of the parasitoids that is giving strong control in Japan. In fact, the strain that we have that showed up here in Georgia is identical to the strain from Japan. So we feel like it came in, we really don't know how, but it arrived, and it is giving us very good control in places. And so we're looking for that population to spread in the near future. When you look at egg masses, you can easily separate egg masses that have been parasitized by the Saccharalis wasp by looking at the color. So the parasitized eggs are much darker in color. The unparasitized have that light color, and that's a little bit obvious there in the image. And parasitism rates here in Georgia are ranging from 55 to 95%, and most commonly above 75%. So this is something that we really feel like is a positive development, but we're looking for that wasp population to greatly expand, and we know nothing about how suitable it is to our particular climate. In the absence of biological control, there are some cultural controls that we're looking at right now. The most effective is planting date. So the earlier that you plant, the more likely your plants are to be infested by the kudzu bug. And our data clearly shows that planting, for example, in April gives us many times more egg masses at the same growth stage as, say, May planted. There's a 2x decrease going from May to June, and more than a 2x decrease than going from June to July. In fact, in July, we had very few egg masses. So photo period and the timing of planting date really is important in terms of managing this from a cultural perspective. There's also some data out of Clemson University that suggests that the infestations are greater along field edges. And there's some things that we can do to more effectively treat those populations without treating the entire field. I'd like to show you just a little bit of data on some things that we're doing with this bug. So the first question I posed then is when does this particular species disperse? And the method that we used to assess that particular response was we operated flight intercept traps. That's just, again, that cardboard vane trap over a black bucket filled with soapy water. And I had 10 of those traps near soybeans or right on the edges of soybeans. I had 10 of those on the edges of kudzu patches. And all the data I'll show today are from here in Tift County. We also assessed the number of egg masses on 20 random kudzu shoots per week, and we looked at that response variable over time. Then the second question that we'll address today is really can knowledge of that local dispersal drive our pest management recommendations? And so we're asking the question, how does timing affect our ability to get more bang for our buck? And we would just want to make one application if we can to take care of the insects without having reinfestation occur. So in that, we put together research plots, and again, this is down in Tift County. And depending on the number that I gave the number assigned to that particular plot, we would spray all the number ones at a given time, and in the next week we would spray all the number twos and so forth as we went through the season, so we could look at the effect of just spraying once at different times. And so that's a single insecticide application using 4.5 ounces of indigo. All right, this is a complicated slide, but stay with me because it's very important. This is 2012 as well as 2013 data. The top slides then are the mean captures. The closed circles are captures around kudzu. The open circles or white circles are captures near soybean. And what I'd like you to see is that there's two distinct dispersal periods in 2012. One of them that's in there about July, and then the second one comes through in November. The horizontal purple stripe is actually the summer solstice. That's June 21, the longest day of the year. And if you'll go down to the bottom graphs, I've got day length plotted in the red line, so that's the number of hours of daylight. You see that on the summer solstice at our location, we're looking at about 14 hours of daylight. And then the black squiggly line is the number of egg masses per shoot. And so up until the summer solstice, we've got as many as 4.5, 5 egg masses, and that drops very, very rapidly following the summer solstice. And so we feel like there's something to it in terms of the bugs shutting off in terms of their oviposition behavior. So I needed to pick a spray date that was going to allow me to get the majority of the bugs with a single spray, and so I just advanced, knowing that the solstice was on the 21st, I advanced that by 3 weeks, and so I just proposed, say, a July 15 spray date would be our best bet, and you'll see how that works out in the data. So this is the single spray trial. Let me set this up. So the blue line is the number of bugs per sweep in the untreated plots. You see the solstice there is one of the purple lines, and then the second purple line is my July 15, which is just my hypothetical date where I think I'm going to be able to get control. So if I don't do any spraying at all, you can see what the population does. On the next slide then, I'm going to show you a July 3rd spray date. So that's a single spray. We knock that population down very, very quickly. But then you see that the population rebounds, and we exceed our 5 bugs per sweep threshold very, very quickly. And so that's not a good treatment for us because we're going to have to treat more than once. If you look at a July 16 spray date, you'll know that there's a strong contrast between the red line and the blue line. So we're able to knock that population down with one spray, and then they stay below our threshold for the remainder of the year. Same thing with the July 31. Again, we see the same thing, so you know the trend here. But this is research, and things don't always work out the way we thought. August 12, I've got a population that I never knocked back down to 0, which is a little bit puzzling, whether we got a rain right after that application or whether we didn't get the material delivered where it should have or mixed properly. I really can't tell you. So August 12 is a little bit of a mystery to me. But if you look forward at the other dates then, so this is the August 20 data as well as the September 3rd data, everything lines up with the exception of that one particular spray date. So I feel pretty confident that we've made great strides in terms of our ability to manage this particular pest in an agricultural context, and that's really based on something as simple as photo period. And so we've got some other things to do in terms of how do we nuance that date as we go farther north. Of course, photo period changes, based on where you are latitude-wise. But the longest day of the year is a fixed point in time. So we're doing some work in different parts of the state this year to try and refine that a little bit more. Okay, so in summary, the kudzu bug is clearly a serious invasive species here in the Southeast and moving to other parts of the country. We see very rapid range expansion across the Southeast into the Mid-South and Mid-Atlantic parts of the country. This insect has got urban and agricultural as well as international trade implications. And I do want to point out that research is ongoing to develop new mitigation strategies for each of the affected stakeholders. This is my final slide. I do want to point out that research, of course, doesn't occur in a vacuum. Got lots of students and technicians that are helping us collect this data. We're very pleased to have funding from stakeholder groups like the U.S. Soybean Board, as well as the Southern Regional IPM project and the Pest Management Alternatives project. And then finally, I do get good support from industry partners that are helping us with crop protection chemicals, and they're very interested, of course, in what we find as well. So from here, I'm going to turn the presentation over to Tracy Leskey, and we're going to shift gears and talk about brown marmorated stinkbug.
Hi, I think, am I on the line? This is Tracy Leskey. Is Willie, can I advance the slides?
Yes.
Okay. Oh, great. Okay, thanks. Hi, everybody. Today I'm going to be talking about the brown marmorated stinkbug and a little bit about the story behind the stinkbug and in terms of the both agricultural and nuisance pest problems posed by this invasive species, as well as some of the approaches we've been taking to start to develop longer-term solutions for the management of this insect. So as Michael was talking about with the kudzu bug, brown marmorated stinkbug is also an invasive species from Asia. It is native to China, Japan, Korea, and Taiwan, and it is classified as an invasive species because of both the ecological and economical harm it has the potential to inflict. This insect is a typical stinkbug species in that we have 5 nymphal instars, the 1st through the 5th. The eggs are typically deposited on the undersides of leaves, and you can see a photo of those there. The 1st instar nymphs remain near the eggs, and then the 2nd through the 5th will move away and start to feed on plant tissue. There really are a couple of reasons why this insect has become such a significant pest. One is that it's incredibly prolificous, meaning it feeds on a number of different host plants. Over 100 have been recorded. And unfortunately, many of these include tree fruit, small fruit, grapes, vegetables, legumes, and ornamentals. And then, of course, the second reason is because it is not a native species, we've seen limited activity from our native biological control agents. And so we haven't been able to keep those populations in check. In terms of distinguishing adult males and females from other native stink bugs, some of the key characteristics include that white banding pattern that you can see on their antennae, as well as the marbled appearance on the sides of the abdomen. The marbling is where it gets its name, its common name, marmorated. And if you want to get some more information in terms of how to separate it from our other native species, there's a very good site on lookalike insects on the stopbmsb.org website. In terms of the history of this insect, it's thought to have been introduced in and around the Allentown region of Pennsylvania sometime between the mid to late 1990s. However, the first official specimen wasn't collected until 2001, and at that point some homeowners submitted some specimens, and those were sent to Rick Hoback at Cornell, who confirmed them, and after that, that was the first official record. In the years that followed, we saw it spread to a number of other mid-Atlantic locations, but for a number of years, we really didn't see any problems from this insect until the latter part of the 2000s, where, particularly in tree fruit, we were seeing this bug coming in the late season into commercial orchards and causing significant injury. In 2010, that was our big outbreak year where it really sort of changed the entire pest management universe in the Mid-Atlantic. And so what we've been doing since that time is responding first to the crisis, and now where we are trying to rebuild our IPM programs with this insect being present. This is a map of the current distribution of brown marmorated in the U.S., along with sort of its level of status as far as a pest. And so there are 4 categories, and currently this insect has been officially detected in 41 states as well as several provinces in Canada. The states highlighted in yellow are those states that have had not only official detections, but also nuisance problems reported by homeowners. And it's a good sign that the bugs are becoming established in those areas. Those highlighted in orange have had recent agricultural problems, and those in red are states where we have seen severe problems, both in terms of the agricultural and nuisance pest problems. Now, back in 2008 and 9, as I mentioned, this is when we first started to detect problems. And I work in tree fruit, and so this is the kind of scene we were seeing back then, where large numbers of adults were coming into orchards and feeding on the crops in the late season. And so for some of our growers throughout the region, even then, we were seeing significant problems. However, by the fall of 2009, it wasn't just our growers, it was also our homeowners, where we had a very large overwintering population. So very much like the kudzu bug, this is a significant nuisance pest where large numbers will come into homes and businesses and even vehicles in attempts to overwinter. And so we knew that we had a large population overwintering, but in terms of its pest status, it at that point had only been a late season problem. However, in 2010, we started to see the bug in the early season, much earlier than we ever had in commercial orchards, where they were feeding on things like Montmorency cherry in this case, as well as newly developing peaches. And because of that, we began to be concerned because, of course, there were cover sprays going on for other key pests, but the numbers in itself were quite alarming. And so by the latter part of June 2010, we started to see the impact from this insect as growers were starting to find significant injury in their peach crops, in their developing peach crops. And what this photo shows is the type of injury we would see from brown marmorated stink bug deep in the flesh of this fruit from this early season feeding. So despite the fact that cover sprays were going on for other pests like plum curculio or some of the leps and even some of our native stink bug and tarnished plant bug, they were not doing a good job for brown marmorated stink bug. And by late July, we were starting to see the devastation as large numbers of growers were seeing significant losses. And in this case, many growers were making the decision to just strip the fruit from their trees and allow it to rot. This was a scene that was repeated over and over throughout the region. And so many growers lost over half their crop, and quite a few actually lost their entire crop to brown marmorated stink bug. Throughout that summer, we really began to understand how devastating this pest could be, as this pest not only affects tree fruit crops, apple and peach, Asian pear, but also row crops feeding on both field corn as well as soybean. They cause that same stay green effect that kudzu bug does that Michael mentioned. They feed on all sorts of specialty crops such as tomatoes, peppers, eggplant, okra. They also were getting into ornamentals and feeding on newly planted trees and orchards, feeding directly through the bark. And they also were feeding in grapes and getting into grapes just before harvest for those destined for wine. And so the issue of taint in wine has also become an important issue for us. So, you know, for our apple growers in the region, just in 4 states, in one crop in 2010, we had about $37 million in losses. So we really had hit crisis mode pretty quickly once this bug had gotten going. And with that, we had a lot of coverage from the press. The press did a pretty good job in explaining the story of this invasive species, and they were interested in the agricultural part. But just like kudzu bug, this bug is a serious nuisance pest. And so this was a photo that appeared on the front page of the New York Times that year, where literally we have homeowners sweeping these bugs up into 5-gallon buckets. And this was not a common scene where tens of thousands, if not millions, were coming into some of these structures and trying to overwinter. You know, one of the things that is absolutely true is that brown marmorated stink bug is a serious nuisance pest, but certainly homeowners, when we explain the definition of a nuisance pest, that it is an insect that does not suck blood or transmit disease, so we refer to it as a nuisance, they are not satisfied with that definition. And in fact, with one homeowner, when I explained this definition to him, this is what he sent back to me in an email. He said, "This weekend I vacuumed up more than 8,000 stink bugs. The vast majority were alive in my attic to add to the more than 4,000 I've removed from my living space since January 1st. I've now destroyed 12,348 stink bugs in my home in 45 days. And he goes on to say that he continues to kill them. And in fact, by the end of that season, he literally had counted over 26,000 stink bugs and took data on it and eventually published a study. But it gives you the kind of scale of the problem and the effects that it has had on the public. But getting back to the agricultural side, throughout the region, what we recognized pretty rapidly is that we had a landscape-level threat to crops and IPM programs. And essentially, this was because we had an insect that was not only feeding and reproducing within our apple orchards, it also was within our cornfields. It was also within invasive plants like tree of heaven, as well as native woody hosts. So in thinking about that, we recognized that we really needed a strong team to come together and address this. And so for the past several years, we have a specialty crop research initiative coordinated agricultural project that we have been working together to solve this problem. And so what I want to talk about now is what we have learned so far, and I can't cover all of the research that is going on. So what I thought I would concentrate on is really the development of some of the monitoring tools and how they may lead to management strategies, because this was a critical aspect for our grower community. So back in 2010, as I mentioned, some of the sprays that were going on were not effective. And so many of us rapidly geared up to screen insecticides both in the laboratory and the field so that we could start making management recommendations, because prior to 2010, only a single laboratory study had ever been conducted with this insect, and so there was no management recommendation in any crop available. So what we had found essentially is that obviously there's a huge range of insecticides, but in terms of having a silver bullet, there was no chemical class that really uniformly outperformed all others, but we did find some representatives within each major class that could be used and be of value in the field. Other things that were really important with brown marmorated stinkbug was that we learned, especially with some of the specialty crops and the insecticide rates that we were able to use, even at the highest doses of some of these insecticides, brown marmorated is very difficult to kill, particularly with a dry residue, because this is an insect that is constantly reinfesting a crop. We also have seen knockdown and recovery, where literally moribund bugs recover from the exposure to an insecticide and start climbing back up into a crop. We've seen that repeatedly in tree fruit, particularly from some of our weaker pyrethroids and neonicotinoids. Residual activity is typically very short. We get about 3-4 days if we're lucky. And so since that time, we do know what we can use, and we've mitigated that threat. But in some cases, the inputs have increased by as much as 4-fold in terms of the number of applications. And also, we've seen frequent secondary pest outbreaks because of the broad spectrum materials our growers have been forced to use. And so a lot of problems in IPM. And so if we look at just, for example, in tree fruit, the insecticides that we could use against brown marmorated stinkbug, what you can see is that we have a range of lethality with those that are pretty good and those that are moderate. These are the better materials that our growers would use. But even residual activity, we're typically talking a very short window of activity, and all of these in some way are fairly hard on our beneficial insects. And I'm not going to even get into the pollinator issues that come with, some of the neonics. But you can see that, the growers really are in a tough spot. And so this is pretty much revealed when you look at some of the spray programs that our growers were using. This is an actual spray program from a diversified farm. And basically every single day they are spraying a different crop for brown marmorated stinkbug, and basically every crop was being sprayed once or twice a week. So this is the kind of thing that we had observed. And so the reason for that, of course, is that we didn't have an effective monitoring tool, a tool that would allow us to assess the presence, abundance, and seasonal activity of brown marmorated stinkbug that we could ultimately use and allow growers to make informed management decisions. So this has been a key part of our research. And so typically in my lab and in the labs of my colleagues that have collaborated on this project, we started thinking about, sort of the key components of any trap-based monitoring system. And typically there are 4. Oftentimes there's a visual stimulus that you can incorporate into a trap design. In this case, it's a black pyramid trap. An olfactory stimulus, the lure or bait, a capture mechanism that is compatible with the behavior. And in the case of brown marmorated stinkbug, both nymphs and adults have a natural tendency to climb up surfaces. So a pyramid trap is a good design, and it allows them to be funneled into a collection device. And because this bug is always coming from the borders, traps put in the peripheral row of an orchard or another crops are are a good place to deploy those. But back to the olfactory stimulus. Prior to 2012, there was one available attractant that you could actually buy. It's referred to as methyl decatraenoate. It's a pheromone produced by another Asian stink bug species known as Plaudia stahli, and it is cross-attractive to BMSB as well as other stink bug species. However, back in 2011, we confirmed that this methyl decatraenoate was not very effective against BMSB because this particular pheromonal stimuli, although attractive in the late season, is not attractive through most of the growing season. So in this case, throughout much of the growing season, the adult bugs do not respond. And so literally during periods of invasion, in this case, late May to early June, where we can walk up to a peach tree, for example, this is where these data were taken, and count 20, 30, 40 bugs per tree, there would be none in our traps. So we really needed to concentrate on trying to find the pheromone produced by this bug. And so this particular result was confirmed in a number of states across the Mid-Atlantic in 2011. And what was interesting at that time was, despite reports in the Asian literature, it did not work as described. We could not observe adults responding to this stimulus essentially until mid-August. So in cooperation with my colleagues at USDA, ARS, and Beltsville, we made a concerted effort to try to identify this pheromone, basically beginning in the laboratory, examining the volatiles produced by this bug, characterizing them, synthesizing them, and then taking them to the field for bioassay. And in late 2011, we made a significant discovery where that stimulus that you see, number 10, the pheromone, that particular compound was indeed the brown marmorated stink bug pheromone that we thought we had identified, and we were seeing behavioral activity in the field. So at this point, we were pretty excited about the response to these baited traps that we were seeing with this number 10 compound. However, it was late in the season, and so the question was, would it be attractive throughout much of the season? Okay, sorry, strange things with the slides, I apologize. This led to trials that we conducted in 2012, where we were asking three key questions. Is BMSB attracted to the number 10 pheromone in the early season? Is it attracted season long? And how attractive is it relative to the methyl decatraenoate, the late season attracted? And so we conducted trials in about 10 states in 2012, and fortunately, we had the ability to do these trials in a very, very unified way, where we were able to synthesize enough of this material to put out experimental lures baited with the pheromone, compared with methyl decatraenoate and an unbaited control. We had new traps that we used and designed so that the traps were uniform, and these were deployed between wild host habitat and agricultural production, and were deployed in mid-April and left in place season long. So fortunately, what we found was that in 2012, with this pheromone alone, we were seeing early season attraction from essentially the time the bugs first left overwintering sites. On the left side, you can see the response of the adults in the early season. In the early season, we obviously don't have nymphs, so there was very little capture. But by the mid-season, as that overwintering population starts to die off and we have nymphs in the field, they are also responding to the number 10 stimulus. The yellow, or the methyl decatraenoate rescue lure that we were using, nymphs do respond to methyl decatraenoate season long. So we expected to see that response, which was good. And then in the late season, you can see the response to both the pheromone as well as the methyl decatraenoate, the rescue lure, which was good. So from there, knowing that we had a pheromone and knowing that we could utilize it, we continued to study this compound, and what we found was that when you combine the pheromone with methyl decatraenoate, that late season attractant, we actually see a synergistic response, where if you would expect to get 2 bugs with the pheromone, 2 bugs with methyl decatraenoate, you would expect 4 in your trap. But in fact, when you combine these, you're getting something like 8. So this allowed us to develop a more sensitive stimulus and led to another broad multi-state trial in 2013 where we compared the response to the pheromone alone and the pheromone in combination with two formulations of methyl-decatraenoate, one at 66 and one at 120 milligrams. And again, these trials are conducted throughout the U.S., and what we saw was very good response to the combination lure, much higher than the pheromone alone. We would see this synergistic response even in the early season. So this was pretty exciting, and again, this worked for both nymphs and adults. And so what we now have is a combination lure that is a very attractive stimulus for the bugs throughout the season. So we really can start to monitor populations season long. And in other trials that we've conducted, I won't present the data, we can also see a clear dose response by increasing the amount of material. You can also increase the number of bugs that are going to show up at your traps and increase the sensitivity. So that's all very good. But of course, the question that then comes up is, can we take this system that we've now developed and apply it and start to develop a pest management tool that a grower could use? And so in 2013, and we're doing it again in 2014, and I apologize, the slides are sort of, I'm not sure what happened in the translation, but basically what we did was to place traps on the border and in the center of, in this case, apple blocks. We're trying to work in an apple system and ask the question, can we use trap captures to guide our management decisions? And what we did was to compare a number of treatments where we would set thresholds for treating these blocks based on trap captures, where either 1, 10, or 20 adults in a trap would trigger a spray application in what we call an alternate row middle pattern, followed by a second application 7 days later, and then the treatment would be, the threshold would be reset. These plots were compared with those that were treated every 7 days or those that were not sprayed against BMSB and were used as a control. And fortunately, what we found, that you probably can't see it on the slides, but what we found was that 10 bugs per trap, sort of, we'll say moderate threshold, was great in that two things happened. One was that, first of all, the trap captures reflected bug activity, and we were able to guide management decisions and also keep injury in the whole plot samples to that we would see with a weekly application, but we actually applied 40% fewer applications. So in the weekly applications, there were 21, and then in the 10 bugs per trap threshold, there were only between 12 and 13. Now, at the higher thresholds, we really started to see it break down, but it tells us from this that we can use these traps to start making management decisions. So we're repeating this experiment in 2014, and so what we hope to do after this second year is really provide growers with a provisional threshold that they can start trying to apply in their management programs and use this as a mechanism to determine whether they really do need to put on one of these materials to, something's happening. Oops, okay, there we go, to see if we need to continue treating this block. I don't see a button to move the slide forward. It's sort of disappeared.
Did it go away?
Yeah. Okay, can you click that again? Oh, wait, I think I got it now. It might work. Okay, yeah, one of the things about the pheromone of brown marmorated stinkbug is that it is an aggregation pheromone. And this is quite different from the sex pheromones we use for moths because there are a couple of key things. One is that you see them responding not so much as a point source that you would see in a lepidopteran trap, but in more of an area response. In other words, they come from a long distance, but they may aggregate around that trap. And so you see them sort of on the trap, around the trap, and then the trees around the trap. And so the question is, can you use that behavior to sort of manage them? And this has led to the development of a potential attract and kill approach for BMSB in apple orchards. And in this case, what we've been doing is laying the groundwork through this through behavioral studies, where what we've been able to demonstrate is that you can attract bugs from a long distance, but in terms of that area of aggregation, it's quite small. You can have a spatially precise location. Basically, the area of activity extends to about 2.5 meters beyond the source. We also see when you put this pheromone out in the context of an apple tree, for example, you can attract and aggregate them within that tree for many hours, in this case over a day. And we did this by using harmonic radar, and you see that little tag on the back of the insect. This is how we can determine if it's still present. We can literally radio tag them and track them through the environment. And then, of course, you need a good killing mechanism. And so we've been able to develop season-long programs for these spatially precise attract and kill sites. And so we've done some quick and dirty trials with this approach and tried different loadings of pheromone and synergist, baiting apple trees, allowing the bugs to aggregate, treating those trees, and then literally counting the bugs that showed up on them, in this case 6 hours and 6 days later. And we're also tracking damage and overall the overall quality of the fruit in these blocks. But what I wanted to show you from last year, we're still sort of in the middle of the season here, well, sort of late season, was that, first of all, you can attract and aggregate a lot of bugs to these sites where we were literally counting tens of thousands of bugs. And with that, we saw a strong dose response. And so what that means is, depending on what we develop in terms of a strategy, a higher dose of pheromone can be used to really attract and aggregate them to the same spatially precise site and take out a lot of bugs. And so we're still developing the behavioral basis for this, but this is one of the approaches we're taking. Now, the last thing I want to talk about before I conclude is just a brief bit about the biology of this bug, because one of the questions we had was, where does this bug overwinter, or does it even overwinter in the natural landscape? This bug is quite prevalent in human-made structures. It's very obvious and conspicuous and highly aggregated. But out in the natural environment, we had no idea if the bugs were present. But at the same time, we were seeing large numbers move from woodlots in the early spring into commercial orchards. So how did we get at this? Well, in this case, we used human surveyors to go out and try to establish if the bug were overwintering, say, in leaf litter or under rocks or anywhere we could think of that the bugs might be present. But in fact, what we found through these surveys that we conducted throughout the Mid-Atlantic was that the bugs were overwintering, particularly in one place, beneath the bark of dead standing trees and also deep within the wood of these decaying trees. And so there could be large numbers of these out in the environment. But in doing that, it took a lot of time. We were able to establish some of the key characteristics of these trees, that they were large, dead standing trees. Particularly, we would see oak and locust with peeling bark. They were porous and dry. But in terms of our ability to survey for these bugs, we didn't cover much of an area. Literally, that little red dot on the right side of the screen was the area we were able to cover over 2 winters. So back in 2012, 2013, we started working with the National Detector Dog Training Center in Noonan, Georgia, to see if we could use detector dogs to help us better survey the natural landscape. And so we literally started working with these dogs, we'll say, in the laboratory, where they were able to establish if these bugs were present in boxes, or if, in this case, non-provisioned boxes, they could differentiate. We then took them to what we would call a semi-field condition, where the bugs were concealed under the bark of trees, and seeing if they could determine if the bugs were present. And at each of these levels, when the dogs reached an 85% proficiency level, we moved them on to a more complex environment. And ultimately, we did take them out into the natural landscape, and the dogs were actually extremely effective at finding these bugs in fact, in the same places the human surveyors were able to locate them, in dead standing trees. So it was a very good survey, but what was interesting is that the dogs could do it 20 times faster. So, this is something we're hoping to get back with them and maybe try to get at the question of what densities are actually being harbored out in the natural landscape as well. So with that, I'm going to conclude by just saying that this project is going to continue, you know, in terms of the way we've been approaching it so far through a cooperative, collaborative, and integrated approach for research and extension. And really at this point, across all of these different specialty crops as well as row crops, everyone's sort of working toward IPM-based approaches, including trap-based treatment thresholds, as I mentioned today, border sprays, cultural control strategies, including host removal, behavioral control strategies. But for sure, all of us agree that, you know, the long-term solution for this bug is biological control, because we really need a landscape-level solution. And so between a combination of conservation biological control as well as classical biological control is being pursued. Now, if you want to learn more about brown marmorated stink bug, you can go to our website, stopbmsb.org, and there is a video series that was recently developed, and it talks about all sorts of aspects of this project, including identification, overwintering and spread, monitoring and mapping, effects on different cropping systems, as well as biological control. So I hope you'll visit our website. And with that, I would just like to thank our funding sources, as well as my laboratory and the entire SCRI team. So I think we're ready to take questions if there are any.
If you have any quick questions, you can type them over in the chat box. At this time, we would like for you to answer these four questions that's popping up on the screen to give us a little feedback on the similar webinar that we had this afternoon.
Should I go ahead and address this question from Sean in Johnston County, North Carolina?
Yes.
Okay, I assume, Sean, if that's for the brown marmorated stink bug, yeah, one of our things that we've been trying to do is get these commercial companies up and running. Now, in terms of our work, it's really toward the development of tools that growers can use. And so there are companies marketing traps for public and nuisance problems for homeowners, but so far we haven't seen any sign that this could be used to trap out bugs in a way that would be meaningful for a homeowner, such that they wouldn't have you know, bugs still infesting their home. So I don't think a trap out strategy will work in terms of for homeowners, but certainly for monitoring this insect, especially in agriculture, it's going to, or even surveillance, it's going to be very good. So if this is for brown marmorated stink bug, Luann, I can tell you that there was a recent study published by Mike Raup that literally tested ready-to-use products that homeowners could use for their garden and rated them for their efficacy against brown marmorated stink bug. So if you email me or if you Google that, I can get you the link to that article, because he did a very nice job rating how good each of these ready-to-use products for homeowners for their garden that they could use.
Dr. Toews and Dr. Leskey, would like to thank you all for this great presentation today. I see there's another question that's come up from Steve.
Okay, Steve, your question is kudzu bug concentrated populations on the edges of fields. Could it be beneficial to plant some type of other crop or barrier plants around the periphery of that field to keep them out? At this point, our experiences with kudzu bug are so new that we have not tried any types of trap cropping with that. I think that probably holds the most possibility for organic growers or somebody like that that's interested in planting something. But I will tell you that one of the different thresholds that we're testing this year is a border treatment where we're using a mist blower and literally just driving around the perimeter of a commercial soybean field and blowing that insecticide in from the outside edge in. And the blower that I have blows about 40 feet into the field. I've treated one field. We had 8 that we were monitoring this year, and I've treated one field that way, and it appears in that case that we got very good control. But I would preface that comment with saying this is very definitely real-world research, and it's not been replicated. So I think long-term that idea holds some promise, but we'll need to get some additional replication. And I've got collaborators in both South Carolina and North Carolina that are following the same protocols. I haven't seen their data yet.
Yeah, that looks like all the questions for now. I don't see anyone else typing. We'd like to thank Dr. Toews and Dr. Leskey today for the great presentation. So if there's not any more questions, that will conclude our presentation for today.